This is episode 81 of the Immunology Podcast, Respiratory Immunology with Dr. Priya Devarajan. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rad. Welcome back to the Immunology Podcast. We have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Priya Devarajan from Stony Brook University on the podcast to talk about her research on immunity to respiratory infections and her recent transition from postdoc to faculty. We've also got our usual roundup of recent highlights in immunology news coming up. But first,
are you modeling the human airway in vitro? Are your cultures physiologically relevant? Can you generate these cultures consistently? Learn more about Pneumocold offered by STEMCELL Technologies, a serum and bovine pituitary extract-free cell culture media for human airway epithelial cells. Expand these cells for extended passages while maintaining air-liquid interface differentiation potential to study respiratory biology, infections, and drug responses. Explore more at www.stemcell.com/pneumacold. That is PNEUMACULT.
are you feeling physiologically relevant today, Brenda?
Always. Are we all not physiologically relevant?
So some days, I don't know. Sometimes I think the planet's telling us that we're physiologically irrelevant.
Well, that's probably true, but what is the fun in that, right?
Well, that's existential dread, right?
Yes, let's not bring it. It's Friday. We're recording on Friday.
Yeah, that's fair. That's fair. And then I head out on vacation. It's LARP time, folks.
LARP time.
7-foot-tall werewolf costume on the summer solstice. Unfortunately, we're going to be under a heat dome, so it's 90 Fahrenheit or whatever the heck that is in Celsius. It's hot. It's like 40-something Celsius, I believe, if I do it right. It's not good.
No, not 40 quite, but very, very hot.
It's almost halfway to boiling on the Fahrenheit scale.
Well, but the scales are not the same. I don't think that's how it works.
No, not quite. 9/5.
But still, way too hot for that, Jason.
Not good.
And how about... And is your wife also wearing something like
not a werewolf
inappropriate?
She just has normal stuff. But I'm also part of the medical staff, and I am absolutely bringing IV saline bags just in case for people.
Oh my God, that's so over the top. All right.
It is until it isn't.
All right. So is this a week long, 2 week long?
Well, Monday, Tuesday are set up. We drive out Sunday night because it's late to get out there. So Monday, Tuesday is set up, goes Wednesday through Saturday. Sunday, pack out. Then we go to Hershey Park for a couple days and come home. So it's a nice 10-day vacation. It's the big family one of the year. Awesome. And then after that, we get the summer shutdown at Ceres. So I actually get the first week of July just off.
Whoa, how generous.
How European of us.
Seriously? Well, that should be August, though. People don't work. The month people don't work is August.
But I've learned that Nestle, which is an interesting development for later, another time, has an August shutdown. So, we'll see how that works out.
Very well. All right. So, very excited about today's research highlights. What do you want to start? What do you have for me today?
Sure. So, in the land of physiological relevance, we're going to hit some gamma delta T cells. This is all about human studies of gamma delta T cells and their relevance here. So this is in Science Immunology. It's titled Human Gamma Delta T Cells in Diverse Tissues Exhibit Site-Specific Maturation Dynamics Across the Lifespan. First authors are Joshua Gray and Daniel Caron, and last author is Donna Farber. And it came out 7th of June. All right. So high level, this is a paper that I'm just going to flag. If you are into gamma delta T cells, you just need to read it because it does not translate well into an auditory description of a story because it's more of a descriptive discovery mapping paper. But what they were able to do is actually get tissue samples from organ donors of lymph nodes and blood and ileum and colon and jejunum, I believe. So different intestinal regions, which are gamma delta rich, blood and lymph nodes, and look across time. And also looked at blood, spleen, lung tissue. And they looked at through mostly RNA-seq, but also T-cell receptor mapping and downstream protein networks. Um, the variable region, the V1 versus V2 delta, and the subtyping, and they're able to really lead up to a couple of things. So one, young kids and baby, as from baby to adult, there's a shift. There's a shift both in the amount that's in your blood versus in your body. There is differences in function, and there's more clonal diversity in the blood compartment in children that is pruned as you become an adult. And there's more tissue repair and stem-like state in children that becomes more effector and cytotoxic function in adults. And so there's a clonal expansion as you become an adult. There's more tissue homing and locking in various resident tissue compartments and compartmentalization. And this is starting to show age-related changes in immunity from kind of a general tissue repair approach to very specific site approaches over time. And that's the large part of the story. They, to understand clonal mapping, right, you have to do TCR mapping. They look at some function where they can. They look at all the compartments. And so it's really a tour de force in data, but then understanding this time component is really what's super valuable, right? Because they're able to take samples that are well enough matched to understand differences between a baby, say 10 to 12-year-old, and a adult. It's not like they just have newborn organ donors or whatever, and 40-year-olds. They have a whole spectrum, and it's a pretty powerful study as a result. So adults age 20, 54 adults age 20 to 81, and 70 children age 0 to 12. So if gamma delta T cells are your thing, this paper needs to be your thing, and hopefully you've already read it. But if not, now you know, and knowing is half the battle.
Indeed. Was there any surprise? Anything they highlighted that they were not expecting?
Not necessarily. It's more that this population's been hard to get at in humans. Mm-hmm. And so it's really been able to understand it and understand the changes. I don't think the changes over time are surprising to people. Like from a stem tissue perspective to a more effector locked in state isn't surprising that you'd have clonal expansion with age. But the fact that you do wasn't even known if that happens with these cells, because getting at it to in humans has been difficult.
Okay, I'm going to start with, what's going to be my second paper, but because there is a hidden gamma delta component,
Ooh.
Let's just, I want to exploit this connection. And so this paper is called An Interferon Gamma Dependent Immune Endocrine Circuit Lowers Blood Glucose to Potentiate the Innate Antiviral Immune Response. And this was published in Nature Immunology, 29th of May. First authors, Marco Shestan and Sanya Mikashinovic, and from the corresponding author, Bojan Polish at the University of Rijeka in Croatia. And for this paper, I thought it was a really nice story and very elegant and a really nice, interesting, overarching story. And basically, they study a phenomena that has been identified, that the glucose levels in blood vary in during the course they focus on viral infections. So we know that when you get sick with a virus, you know, this idea of feeling sick in partly is because of the differences in kind of nutrients that are, so you have cytokines that make you feel sick, but also differences in the nutrients in your blood. And this is part of the whole syndrome of being sick. And one of the nutrients that definitely changes is the availability of glucose. And so one of the things is that you usually have lower glucose concentration, and depending on the infection, so they discussed that in some levels, if you have a very severe infection, then you have higher glucose levels. But in their system, you have a kind of strong infection, but not lethal, not too severe. You actually have a lower glucose concentration. You have lethargy, reduced appetite, and that this is observed in humans. And It is unclear what is the connection between blood glucose levels and the antiviral response. So and this is particularly important in the case of people with diabetes, which we know have uh, un, unregulated amounts of glucose and usually higher glucose levels. And people with diabetes usually are known for having heightened susceptibility to viral infections, and so they're trying to find kind of a link between these things that we know. And they use this mouse model of infection with a murine cytomegalovirus (MCMV), and they measure basically glucose concentration, and they um, they mimic different doses of viral infection, and they observe that in both female and male mice, there was a drop in glucose concentration at certain non-lethal or fairly high amount of virus. And this was shown to this was shown to como enhance the antiviral response, and they see the decrease in blood glucose concentrations, both male and females. So this reduction in glucose concentration that they see in mice, which mimics what they see in humans, and they use this as a model to study where is this glucose reduction coming from, and what is the implications for the viral response. Basically, they do some in vitro experiments, and they show that indeed less glucose uh, improves the antiviral response, and but what they show is that this is by restricting glucose metabolism. They restrict the production of lactate, and this actually enhances the production of interferon beta, so interferon type 1, which in turn increases the antiviral response. So basically, they see less glucose, less lactate, and through inhibited activation of IR3 and the canonical NF-kappa beta pathways, they uh, end up reducing the interferon beta production. Sorry, enhancing. Sorry, if you have less glucose. So what is and by this is in a method, and it shows also in vivo that is a method of enhancing antiviral response. And I thought was very interesting is that then they try to understand why is there a fall in glucose concentration, and basically what they show is that there is a increase in insulin production in the pancreas, and this increased insulin production in turn is initiated by an increased production by basically by the effects of two cytokines. On the one hand, they have interferon gamma that is increased in plasma during this infection, during the early stages of the infection. And then after some kind of work around, they also realize that this interferon gamma coordinates with IL-1 beta that is produced not systemically, but by myeloid cells, macrophages that are either colocalizing in the pancreas, are migrating to the pancreas, or are in the pancreas, and that combination of two cytokines activates the pancreatic cells to produce more insulin. And here's where the gamma delta connection comes, because the source of this interferon gamma turns out to be gamma delta cells in the spleen that get activated when the virus kind of is at high enough concentrations to penetrate in the spleen and gets and activates the gamma delta cells that are within the splenic red pulp. And this is kind of the threshold that activates this whole cascade that ends up reducing glucose availability and increasing therefore interferon beta production. So I thought it was really nice, a really cool kind of sequence of events, and basically a very interesting mechanism by which you connect, the level of viral infection with this metabolic um, mechanism.
That's super interesting because glucose levels in various fluids are used to identify bacterial versus viral infection, for instance.
Okay. All right. Oh, I just want to say that the way then they add a model of diabetic mice, then they respond worse to this, to the infection, and in part is because of the glucose, their how they fail to respond to either produce insulin or respond to the infection by reducing glucose availability.
Interesting. All right. Well, I don't have a segue to glucose here. But we know infection, inflammation. So this paper made the rounds on the Scienceverse. I saw all over the Twitters, or X's as it's now called, and the LinkedIns. So all the socials. So it's titled "A Disease-Associated Gene Desert Directs Macrophage Inflammation Through ETS2." It is in Nature, coming out on the 13th of June. First authors, C.T. Stanky, C. Borges, and L.M. Hong. All were first authors. And last author is J.C. Lee. All right, so this is a real tour de force of some genetics analysis. I will say the news is publishing it as "Group Finds Secret Master Driver of All Autoimmune Inflammation." Blah, blah. It's a little oversold, but it's still actually a really cool paper. And we know that when scientists and we all talk to media about our papers, that then someone, at Reuters or whatever converts it to be a little more exciting than it maybe is. It's still exciting, but we can keep it at nerd level excitement, not cure for all immunity excitement. That being said, there is this known genetic spot on chromosome 21q22 through GWAS and other studies have been shown to be independently linked to inflammatory bowel disease, ankylosing spondylitis, primary sclerosing cholangitis, and Takayasu's arteritis. So what's going on there? What they do is they identify a gene that's a driver of this, but how it works is where the really cool science starts to get in. So what they find is that there is a spot on the chromosome that is a super enhancer. So it's downstream. It's not in a coding region. But they looked for nearby genes lacking that have been previously studied, which include ETS2. And so they used promoter capture high C and expression quantitative loci data and a bunch of other fancy techniques. They identify ETS2 as a culprit here with polymorphisms. They then understand what this polymorphism does, and it serves as a super enhancer to pull in the pioneer factor PU1 into the bound DNA to serve as a super enhancer. So this SNP leads to more ETS2 in the end. So that's the first part of this paper. So what the heck does ETS2? Well, ETS are families of transcription factors or proto-oncogenes, but they do in human macrophages is not well known because cell lines or mouse data have contradicted each other, and they explain they give the whole reason, which are a good set of reasons about why this is weird. So they take human macrophages and start doing CRISPR, and they knock it out, and they see less inflammatory potential in them, less ROS, less phagocytosis by assays. So knocking it out is bad for macrophage effector function, right? They did RNA-seq and saw all the cytokines go down and everything else. They don't see they don't see changes in chromatin accessibility driving this, but it looks like it's a just generic transcription factor. And they also codify a similar effect in the genotype that's associated with this chromosome region in IBD patients. They can see the macrophage function in IBD patients with this known genetic type. So then they look at overexpression. So they did a specialized overexpression system and see activation of the function. And so they then look at GWAS studies and focused on IBD because that's where the most data is, and tried to understand, well, given what we know and we think it's this, can we remine the data and see if there's linkages? And again, they see the same story coming out in human data if they know where to look, right? in GWAS of people with a genetic predisposition, and they see the ETS2 signal there of the downstream. And they also see this with all the other conditions, PSC, ankylosing spondylitis, Takayasu's arteritis. They identify specific inflammatory effects and drive down. They look at oxidative phosphorylation and glycolysis. But just rescuing those doesn't rescue the function. So those are more downstream effects, right? So it's not just an immunometabolism story or an OXPHOS story. It's fundamentally altering the entire transcriptional profile of the cells. And then they use a small molecule and show that that can indeed have an effect in systems and in animal colitis even. And can then compare that to anti-TNF effects. Is a kind of a model system. So high level, there was this, known chromosome SNP popping up on GWAS, not in a coding region. They identify it as binding a super promoter, a pioneer factor. They identify that as activating ETS2, and they figure out that ETS2 is a master regulator of macrophage function writ large. So when you have more of it going, you have more macrophage activation, which pan macrophage party leads to more autoimmunity. Thank you, Nature paper. Goodbye.
What a ride. Very interesting. I mean, I really like these GWAS papers that end up, hitting something good. It's very satisfying, isn't it?
Yeah, well, this is all known GWAS data. They were trying to make sense of it. And so they really did some sophisticated chromatin mapping and other work to figure it out. I was very impressed.
How big is this effect? Because it feels like this.
It's enough that there's functional downstream effects in the macrophages in vivo and in vitro and in vivo models, right? And they see it in patients, so they see this specific pattern appearing in sick people.
Yeah,
but it's everything, right? So it's not just like, oh, this one cytokine's up in macrophages. You have significant bumps in all function with upregulation or decrease in function with downregulation.
yeah.
So that's why they're billing it as a master transcription factor.
Yeah. I wonder that if you're doing experiments and you're like, oh, the whole macrophage is messed up. I don't know. There's something. The experiment went wrong, or this donor is weird, and they just ignore it.
Maybe, but like that's why they picked this chromosome region, right? This was known in all these diseases that keep popping up as a signal, but not why. So there was the smoke, and they finally found the fire.
All right. Okay, so for this last paper of the day, I have I don't know, I'm not sure how to feel about this paper. I thought it is kind of a interesting thing. Sometimes I feel it was quite a long paper, and then I think the story is fairly straightforward, but not really well. Let me just walk you through it, and you tell me what you think. So this paper is called "Cellular Architecture Shapes the Naive T-cell Response," published in Science on the 7th of June. First author Benjamin Hale, corresponding author Berend Snyder at the ETH in Zurich, in Switzerland. And this kind of the whole premise of this paper kind of revolves around a quirky observation. I guess that's how it started. Um, that there's this some T cells like naive. You look at the naive T cells from blood. A bunch of these cells, if you stain them with the right thing, have a kind of a weird shaped interior. So they start the publication by they want to understand what is the origin of the heterogeneity that we see when activating naive cells, right? So within a naive cell population, you're going to have some cells that even if they are exposed to the same stimulation, even if you have cells from, transgenic mice that have all the same TCR, you're going to have some cells that are going to end up differentiating into, the effector cells that guide the response. You're going to have memory cells. Some cells activate faster than others. And I think there's still a lot of kind of un- lack of kind of clarity around where does this variability come from. And there's of course, extrinsic reasons for the cell, that have to do with sometimes you have more peptide presentation or certain, the Some cells get more nutrients than others, things like that. But there's also some intrinsic reasons. Some of them are more like stochastic and like that have to do with, variable expression, particularly of TCR and core receptors. If you see them when you stain them, there's like a little bit of a smudge. Some cells have more on their surface, and cells have less. And there's probably no real reason for it. But they were really trying to find like non-stochastic contributors to this heterogeneity. And so one of the things they define, and this is something that they had already published kind of to some extent before, is what they call T cell architecture. And this is where the shape comes into place. They show that if you stain, human PBMCs and they focus on the CD8 cells, you stain them with intracellular anti-CD3, you see kind of a weird pattern in which you have, on the one hand, you have they have kind of they show that you can divide the cells in basically three rough groups. On the one hand, you have cells that what is they called polarized, which is basically cells that are not round. So this is often you'll see some cells that are kind of a little bit elongated, and this is especially this is what you see mostly also when you activate naive cells. They start kind of shifting shapes. But then you have, and those are kind of a minority of the cells, but then within the nice round pretty naive T cells, they identify two distinct architectures that they show are fairly consistently found across donors and across circumstance. One of them is what they call just kind of the conventional T cell, in which you just have the homogeneous T cell surface with CD3 distributed around, and then you have a different type of cell that has this kind of CD3 expression that is proximal to the nucleus, and it makes this line that goes inside that looks like it's going into the nucleus. And it looks, so they call it stripy, and it looks like, what would be the symbol like that you can use for diameter, how what would be the Greek, which letter would that be? Kind of the circle with a little like cross, a cross with a line.
Pi is the
Kind of. Yeah, like the but that would be the like the yeah, the big capital pi, right? Or phi. Phi, not pi, phi. So they have it has this weird shape. And so basically an invagination with that kind of grows into the nucleus. And then they have a lot of very fancy microscopy, and they use some, they use AI to like analyze thousands and thousands and thousands of cells in different slides, and they show that this is something that happens across the board. And they go ahead and try to understand where these cells are coming from and whether this means something biologically. And I think this is what it gets kind of interesting. Until now, it's just kind of this observation. It's quirky, great. So they go through a lot of lengths in characterizing these cells. They show that not only do they have like the CD3, but they also have uh, other components that are kind of concentrated around this invagination next to the nucleus. They have mitochondria, ER, nuclear pore complexes, and they have this whole structures. It's a thing. It's just a structure that's generated inside the cell, and that they are fairly abundant within kind of these naive CD8 populations. And this is seen both kind of in human cells, but also in mice cells, and also within transgenic mice with transgenic TCRs. They also see them OT1 mice, P14 mice. And I think the most important conclusion that they reach is that these cells have a different tendency when they get activated to differentiate compared to those kind of nicely round, non-topologically kind of clean T cell structures. So the T cells that don't have this stripy phenomena. And that this kind of long story short, they do a lot of characterization and a lot of showing that these are kind of distinct subset of cells that are different to the other ones, and they're primed to become effector cells. They proliferate more if you kind of pick up clones that look like this and you activate them and you compare them with clones that are round and homogeneous. They have a lesser tendency to differentiate into effector cells that proliferate very quickly. And they think that this structure that they have might be priming them towards responding faster. But these are naive cells, they're not memory cells. And that they kind of they kind of conclude or they propose that this different, what they call cellular architecture, is actually an important feature that differentiates cells, and that primes certain naive cells towards a more quickly activation. And this might explain part of the heterogeneity that we see when we activate naive cells. And on the other hand, the conventional round cells with no features, those ones seem to have a higher tendency to differentiate into a more memory phenotype. And in part, this is mediated by differences in their calcium signaling because the stripy cells can release calcium faster and respond faster to stimulation than the round cells without features. So I don't know, I thought it was a really they do a lot of analysis, but to really show that these are kind of that this architecture is something particular and it's not just an artifact that they're observing. They see them in human cells, in mouse cells. They see them across the board at similar amounts. So the percentages that they see are kind of comparable across donors and across mouse and humans. And that this does affect the existence of the structure affects the early stimulation of the cells and has a kind of functional consequence to how they end up differentiating.
So do they know why the shape is different and if that causes the function, or is it just like, hey, they look different and do something different?
Well, because the shape, what they see is that they have this all accumulation of ER, endoplasmatic reticulum, and mitochondria, and like kind of the nuclear pores. They have all these things kind of in one place, and that's what in this stripe, and that's what generates the visible structure. And in this way, also, it seems to change the way it accumulates calcium, and that is probably where the functional effect comes from. That because of the structure, they have a different kinetic when it comes to calcium release upon stimulation.
Got it. So it's the ER that's being picked up as the gross shape that you would really only appreciate on EM, but you can see is perturbing it on regular.
Yeah. Well, they also, so what is funny is that they see this if they stay with intracellular CD3. That's the first thing they saw. And then they do a lot of other measurements, including ER, including mitochondria, and they see that there's all these other structures that are also co-localized in this stripe, in this central stripe.
Well, our conversation is about to co-localize here in a second with Dr. Priya Devarajan at the Stony Brook University. But before we get to that, this week, fitting nicely with our guests, we'd like to introduce Pulmonary Cell News, covering everything long from COPD to CF to cancer. Pulmonary Cell News keeps readers current with the latest news, research, policy, events, and jobs relevant to the lung research community. Check out Pulmonary Cell News and the rest of STEMCELL's scientific newsletters at www.pulmonaricellnews.com.
So today, for our second part of the show, we will, as always, have an interview with a new guest. Joining us today is Dr. Priya Devarajan. She is an assistant professor at Stony Brook University, brand new, started this month. And she's going to share with us some insights about her research on respiratory immunity and also some career insights, especially her recent experience applying for her tenure track job. Thank you so much for joining us today, Dr. Devarajan. Welcome to the Immunology Podcast.
Thank you, Brenda. Thank you, Jason, for having me here. I'm really excited to be here. I hear a lot of your listeners are students and postdocs and, the next generation of scientists. And I'm excited to talk to all of you.
And you're recruiting, right?
And I'm recruiting. Yes, guys, join my lab. It's amazing.
I know at least one case, or if I may just say, I know at least one person that decided to join a lab after hearing the conversation with in the Immunology podcast. So, this is the place to be.
Perfect.
Right, Jason, go ahead.
There's going to, I think, be a good talk of the career path and how you got to where you were today. But let's start with the science, and that's why someone may want to join your lab too, with COVID and lung inflammation and kind of what is your work to date, and what are you about to go into with this field, given everything that's happened with everyone's lungs since 2020.
Yeah. So I started, I've been fortunate that ever since the beginning, ever since my undergraduate lab experience, I've always wanted to work on immune memory, really asking what helps your immune system remember an infection to come back stronger next time and help clear the infection. And so during my PhD, so I was looking for infection and immunology labs, but I somehow found this really fun new PI who had started his lab, and he was doing type 1 diabetes. So he said, you know what, we'll design a memory project for you. So during my PhD, I studied autoimmune memory because, I had to do memory. But it was pretty cool because it was a fairly new concept at that time that your immune response can also remember self-antigen. And I was studying type 1 diabetes. So in this case, it can remember pancreatic islet antigen and come to attack the pancreas. And memory is going to be harder to suppress than an effector response, for example, because normally when your T cells get activated, they become effector T cells. These effector T cells go on full force, help clear infection, help clear the foreign pathogen, and then they will contract to become long-lived immune memory, which is more of a quiescent state. But then they're well poised to come back and fight the next infection even better and faster because you've learned how to do it the first time. So in autoimmunity, this is, of course, a bad thing because you don't want your T cells or your B cells or any immune cell attacking your own organ. And so if you have memory and immune memory against the organ, that's definitely not a good thing. So during my PhD, I studied autoimmune memory. I studied how this molecule called CTLA4 regulates autoimmune memory. And that was really cool because it was a whole different angle of immunology. And then for my postdoc, I went on to do immune memory against infection, which I always wanted to do. And how that happened was in my PhD, because I was a big fan of immune memory, I invited Dr. Susan Swain to come and give a talk as a graduate student. And then I followed her all around. I'm sure she must have been like, who is this PhD students hanging out around me all the time? I followed her all around, convinced her enough to take me in her lab. And that was a very fun postdoc experience. During my postdoc, I studied T-cell immunity against respiratory infection. I also got the opportunity to do a bit of B cells because the lab did both T and B cells. So I expanded my horizons to also include the other adaptive immune arm. And so during my postdoc, I studied not really the memory part of the response, but really the effector part of the response. So in the past decade, what we've realized is that it's not just that you have T cells and B cells, but that you have different kinds of effector T cells. And of course, we always knew that. We knew there was CD4 T cells and we knew there were CD8 T cells. And classically, we thought the CD4 T cells were the helper T cells. So they help the B cells and they help the CD8 T cells and all the other immune cells mount effective immune responses. And so what we've learned over the past decade is that these CD4 T cells themselves can be specialized, beyond the whole Th1, Th2, Th17 paradigm. They can be functionally specialized. So they can be functionally specialized to help B cells mount successful antibody responses. And this subset of CD4 T cells are called the T follicular helper cells. And very interestingly, CD4 T cells can also kill. It's not that just the CD8 T cells that can go around killing pathogen. CD4 T cells can also do that. And that subset of CD4 T cells are called the cytotoxic CD4 T cells. And so during my postdoc, I tried to understand how these T follicular helpers and cytotoxic CD4s develop during an immune response against influenza, and how do they successfully mount an immune response to clear the virus. And so in my PNAS paper in 2022, I published the requirements for the development of these T follicular helper cells, an aspect. Of course, we know a lot about how these TFH cells develop, but further insight on how they develop. And last year in 2023, I published a Cell Reports paper on how cytotoxic CD4 T cells develop during influenza infection to be able to clear a virus. And now, as I start my own lab, I'm really excited to study respiratory immunology. So over the past 2 decades ago, when people studied immune responses, they were looking at immune responses in the lymph nodes, in the spleen, maybe in the blood. And over the past decade, we've realized that every organ is really an immune organ. You have immune responses in the lungs and the kidneys and the skin. You have immune responses within the tissue that's infected. But we still aren't as clear about all immune responses that go on in the tissue, how they are supported in an organ that isn't meant to be a lymphoid organ, right? Because lungs are supposed to help you breathe. So, how does the lung mount an immune response? So, we're understanding more and more about tissue immune responses. Um, so, that's going to be a focus of my new lab. Studying how successful immune responses are made not only in the lungs but also in the nose, and that's something the whole COVID pandemic spurred on as a whole field is nasal immunity. So up till now, most labs were studying respiratory immunology, at least the basic science labs were studying lung immune responses. But it's interesting to think about it because when most of us lucky, healthy people, otherwise healthy people, get infected by a respiratory infection, like the flu, for example, it's really our noses that gets infected, that our upper respiratory tract that's infected, and it's cleared. And if the infection goes far down as the lungs, you're likely in the hospital. Um, so if most of us are mounting immune responses in the nose to clear respiratory infections, then we really need to know more about nasal immunity. But we know so little. And I guess a large reason of why that was because it's really hard to study the basic science behind it because we study basic science in mice, and mice have really tiny noses. But now we have lots of tools and methods to be able to do that. So I'm really excited to study nasal immunity and how nasal immune responses can help clear respiratory virus. I'm also in another project, I'm also studying how tissue resident memory cells form. That is, if you have an immune memory cell in the lung clearing virus, where does that cell really come from? What kind of effector T cell makes this long-lived, long tissue resident memory cell? And so that's one project. And to sort of give you a quick look into our preliminary data on that, what we're finding is that these cytotoxic CD4 T cells, which are CD4 cells that can kill virus directly, actually become lung tissue resident memory. And it looks like they're a major precursor of that, which is pretty cool to think about it. And in a third project, we're studying immune responses as people age. If you think about, again, who are the people who are most impacted by be it flu or COVID, it is older people who aren't able to mount strong responses with a vaccine and who are very susceptible to these respiratory viruses. And so we try to ask what makes a successful immune response in an aging immune system. And what we've found is that it's not the same thing that makes a successful immune response in younger people, is that there again are specialized aged subsets which can mount successful immunity. And maybe we should be thinking about how to target these subsets to specifically make vaccines for the aged, instead of trying to give the same vaccines that we give to young people. And so that's the third component of, again, same thing, respiratory immunity, but respiratory immunity in aged humans. What can we do? And aged immune systems, really, because mostly we use mouse models to answer the basic science questions.
Wow, that was there's a lot of plans I see for your upcoming research, and I think I am particularly very interested on cytotoxic CD4 cells. And I always, when I see them, I think this is fairly more recent that we're starting to understand how the roles that they actually play in immune responses. I think also in cancer. So I want to ask, like, a very basic question, but I think that maybe some of our listeners might also be wondering, how do these cytotoxic CD4 cells act? Because when you think about it, textbook immunology is MHCD4 cells are recognized as MHC class 2, and that is expressed on only on selected cell types. So what is the missing link here to understand how these cytotoxic CD4 cells are killing virus infected epithelial cells, immune cells? What are the targets that these cytotoxic CD4 cells are recognizing and being cytotoxic towards?
So I think we could start answering that question by first asking, why do you need cytotoxic CD4s? We have a whole arm of CD8s, right? Which are out there to take care of killing pathogens. So why do you even need CD4s? And that's because a lot of viruses, again, both influenza and, as we learned over the last couple of years, also SARS-CoV-2, can downregulate class 1 as an immune evasion mechanism. And so if you have class 2, and if you had a class 2 dependent killing mechanism, there you have to combat immune evasion by the respiratory viruses. And so during influenza infection, for example, respiratory epithelial cells will upregulate class 2. And so this way, our CD4 T cells can see that a cell is infected and can go on to target these cells. So, in short answer to your question, infected cells can upregulate class 2, and CD4 cells can see that and kill them. And that's an important way to overcome immune evasion by these viruses.
So as a follow-up, looking at your lab website and what you're working to, you mentioned nasal immunity. We had that one live attenuated flu vaccine, which had some issues because it was too alive. But then we've seen a slew of papers coming up about, hey, look at this great nasal immunity for COVID if you do it this way. We've covered several of those on here. It looks like you're starting to study this as well. Why hasn't this been just adopted by everyone and their uncle immediately based on the data today? Do you have any insight into that? Because it seems so obvious that that's what we should be doing. And since we have mRNA technology, we could have done it yesterday, but we just aren't.
So I think a big reason for that is because of what we had up till now as our only example, pretty much, of an intranasal vaccine was the flu mist, as it's called in the US, which is a live attenuated influenza vaccine. And so for listeners out there who don't know what this is an influenza vaccine which contains influenza virus, but then it's attenuated. It's attenuated so that it only replicates in the upper respiratory tract. So it's grown at a lower temperature, so it can replicate at a lower temperature in the nose. And so that's how it's attenuated. And so I think a lot of intranasal vaccines got bad rep because of the flu mist, because there have been many years where the flu mist didn't offer as good protection as our intramuscular vaccines. But what I guess the general public doesn't realize is that they didn't work not because of a failure of intranasal immunity, but there were other reasons that it didn't work. For example, none of this is set in stone as we know exactly why it didn't, but one of the potential reasons is because of viral preparation. If you made live virus the same way you make live virus that infects the lungs, it's different. You have to prep it differently for the nose. And so it was a problem with the viral prep itself for the live attenuated vaccine that we think might be a reason why flu mist did not work certain years. Another reason is simply because the viral strains aren't as well matched to the circulating influenza that year. And so the viruses that were made that year for the flu mist weren't exactly what was circulating that year, so they didn't provide good protection. So I think as a field, we should not use the inadequacy of flu mist during certain times as our argument against intranasal vaccines, because there's a lot more to that. like I just explained, a few reasons out there. And there's an excellent review, I think, in Lancet that was published a few years ago that you could read if you're interested in live attenuated virus vaccines, a flu mist, and reasons why it might have not worked as well these years. And I think there's also been, because there's not much enthusiasm for intranasal vaccines, there's been a delay in the technology to make these intranasal vaccines. You can't, in terms of vaccine delivery, again, you have to adapt it to delivery for the nose. You can't produce intranasal vaccine the same way as you produce intramuscular vaccine. That's something vaccinologists are working on to deliver this and to have the right formulation for that. And again, I think it's because we didn't, we thought, okay, flu mist doesn't work as well. Why try? And then COVID came and we made the intramuscular vaccines. And then we started doing the basic science of these studies and seeing that, again, there are nasal immune responses. Maybe you should give this another shot. And so I think the whole field is getting another rebirth by COVID. And I think, I expect to see good intranasal vaccines in the market because I think nasal immunity, again, I said, just looking at successful immunity against respiratory infections, a lot of it is nasal immunity. And I think that's also where I try to sell the work that our lab tries to do is a lot of us are trying to make intranasal vaccines, but there's a big gap in knowledge about the basic science of nasal immunity. And that's what my lab hopes to do is if we see nasal vaccines are working, how exactly are they working? Because nasal immune responses are different from lung immune responses. There are different epithelial cells lining their nose. There are different antigen-presenting cells in the nose. These are all different subsets. So there's a big gap in the basic immunology of nasal immunity. And that's a gap that my lab is going to try and fill.
So when it comes to studying nasal immunity in humans, and I guess, you've already written some grants about it. How do we do this? Because it's not like the mice where you can just, get all the nose epithelium and just study. How do you access the nasal immunity in human subjects?
So I'm going to start with a disclaimer that I'm not an expert in human immunology, so I might be saying something very naive. And if I am, all the listeners come back and give me feedback. But my understanding of the field is that nasal immunity is actually one field where we might have more human data than mouse data, because people have been doing nasal swabs for some time now. And when I am, going out online looking at papers, trying to write grants, I'm able to find more human nasal immunity papers than even mice. And so I think nasal swabs are an excellent source of human nasal tissue. And so I think it's another reason why the field is likely going to move faster in terms of translation and the link between linking mouse basic science studies to what we see in humans, which is also why I think all of the basic science mouse immunology research that we'll be doing in my lab, we're going to try and follow up with human nasal swab samples that we will have access to. So I think it should be easier than a lot of other tissues to study human nasal immunity.
So switching lanes a little bit here, you just started a lab, which means you successfully got a job offer as an academic. So I think for many of our audience, including maybe someone else on this call, Brenda, would love
asking for a friend.
Do you have some advice on how one goes about and does that successfully?
I think it's important to remember that and to tell ourselves that everyone's path is their own. What I heard the most when I asked experienced people, who are already PIs, and I said, hey, how do I become a PI? The most common answer I got was get a Cell Nature Science paper, get a grant, which is really daunting to hear because it isn't easy to just go out there and whip out a Cell Nature Science paper. And I think, unfortunately, that's not how science should be defined. I think there are plenty of excellent papers out there in the Journal of Immunology and, many other very good journals. But that's what you get told is you need those big papers and you need this grant. That's how you get an offer. And that's really off-putting for a lot of people. Because like I said, everyone's career trajectory is different. Everyone has chosen a postdoc very differently. When I was looking for postdocs, one thing very important to me was that I needed to be able to enjoy my science. I wanted to be in a great environment where I could do science because I loved doing it. And so I wanted to join Susie Swain's lab because I'd met her during my PhD, and I'm like, this is where I'll fit in. And I love working with people. I got opportunity to mentor students and so on. So my path was different. Everyone's path is going to be different, and don't stop yourself from trying just because you didn't match these two things that people say is required. Do your best. Have fun doing your best. See where it goes and apply for grants you want to apply to. Do not limit yourself because you think I'm not good enough or because you think, well, I don't have enough data to apply for this grant because, I didn't discover this huge thing. No, go out there, write down the grant, enjoy writing the grant, put it out. You don't know if you're going to get it. That's how I got my grants. Last year, it was very interesting because once I had the attitude of just brute force, just saying, you know what, I'm going to do it because I want to do it, because I like doing it, and I'm just going to give it all a shot. I sat down, I wrote grant after grant after grant. I did everything I wanted to with my papers. I got my papers out and I got those grants. I was very surprised. I remember checking my R21 score and looking at the score and being like, what? Am I actually going to get this? This makes no sense. Someone actually trusts me to give this money. So I think a lot of us tend to underestimate ourselves, underestimate our ideas, and we find it hard to judge ourselves. So my biggest advice is do your best, do what you want to do, have an idea of what's required, which is you're required to have done good science, you're required to have published that science, and you're required to have had good ideas that you can put into grants. And go out there and do it. Just do it. Don't waste too much time thinking about can I, should I, am I good enough? Not worth it. You'll never be able to answer those questions. A lot of people, I feel like, especially women, minorities, people who didn't grow up seeing role models, didn't grow up seeing, their family or people that they knew as scientists, we think it's this goal that's impossible to reach and we don't even try. And so I think that's also what puts us back, that just not thinking that we can do it. And I think that's why also diversity and equity and inclusion is so important because people need to see that there are scientists that look like themselves out there. This is something that you can do if you want to do it. So really my biggest advice is choose science that you want to do, that you think that's fun doing, that you can put your heart into doing. Have fun doing it. Get feedback from everyone constantly for your science as well as for your career. But understand that feedback should be adapted to yourself. And again, for your science as well, everyone's going to give you lots and lots and lots and lots of ideas, but your project, talk to your advisor, talk to your direct mentors, see what you can apply to your science. For your career, again, you're going to get lots and lots and lots of advice. And I think that was one thing I did right. If there's anything I did right, that was one thing, is I asked everyone I could find for advice. And I listened to it all. It was all very different. People sometimes told me opposite things, but listen to it all and adapt it to yourself.
So just go for it and get all the feedback you can get. I think that sounds very reasonable. And just also particularly for our female listeners, you did interview for your positions pregnant and it worked out for you too. So maybe I think this is also something you might want to share with our audience. How did that work out for you?
And this is something I have spoken to a lot of women about, postdocs especially. A lot of us are older and older as we're doing our postdocs because, the requirements for publication are becoming harder and harder. So we're spending longer times in graduate school. We're spending longer times in a postdoc. And I know a lot of women like me said, you know what, I'm going to wait to have the baby. But sometimes you keep waiting and you're like, okay, how long am I going to wait? At some point, it's going to become biologically difficult to have that baby. And it's a lot of, again, prejudices that you have in your own head, um, that people have told you, that you've heard, that you've seen, that stop you. You think it's going to be really hard. People are not going to give me a job if I'm pregnant. Or if I'm pregnant, I'm not going to be physically able to do it. Oh, I'm going to have baby brain. My head's not going to be clear. Again, my advice is do it. If you think you're ready, if you think that you want to do it, and things will fall into place. I think there are a lot of myths out there, which I had an amazing gynecologist. So, again, if you want to get pregnant and if it's possible for you to talk to a few gynecologists, do it. Because having someone, and my gynecologist, again, to give a shout out to her, it was Dr. Gina Sullivan at University of Massachusetts Medical School. And she was someone who she told me her story when I was worried. I said, you know what, and I remember meeting her in my first trimester. I just started applying. I was applying for both grants and positions and doing revision experiments from the paper. And I said, I want to do it all. I'm pregnant. I don't know what to do. And she said, you know what, do it all. I did it. I applied for my residency in my third trimester. Things were very chaotic. Well, you'll figure out ways to do it. And I said, you know what? Am I going to have baby brain? Am I not going to be able to think on my feet? She's like, no. But what people don't realize is that you're a woman who's been doing science for so long. So what might sound stressful to a lot of people out there is your everyday life. You figured out how to do it. You figured out how to make this possible already. So maybe this will come more easily to you. So don't let anyone out there stop you from interviewing because they say, oh, it's going to be, your brain's not going to be clear. In fact, interviewing when I was pregnant worked out well, because I didn't get much sleep. And frankly, I don't know. The brain just adapted itself. I can't think of a time when I was in interviews not being able to think straight because I was pregnant. Your body mechanisms kick in. It's again a wonderful system that's learned to work, with all the other organs. You don't realize how your body can adapt to things. So don't worry about baby brain. I was worried about whether I would be physically fit. But again, I kept walking. I kept doing experiments that I was doing. I found ways around if I got more hungry. You find ways. I found ways to take a snack. I found ways when I need to take a break. I found ways to go on interviews, be on my feet for 10, 12 hours. Like my gynecologist told me, you know what, maybe you can take the next day off. Go get a massage the next day. And it was not that bad. So don't stop yourself. Most of what stops us is in our own head. If you're ready and your career is ready at that point, go ahead and apply. And I think one of the best pieces of advice that I got last year is, what are you going to lose? Right?
Yeah.
Apply, go on the interview. Worst case scenario, you won't get the job, but you wouldn't have got the job if you didn't apply anyway.
Yeah.
So do it and be happy doing it. A lot of people are going to tell you a lot of things, and take what's going to help you and tell yourself that I can only do my best. And so I'm going to do my best and I'm going to let go of everything else. So I'm going to give it a shot, give it my best. I'm going to try my best to have fun while doing it, or else it isn't worth it. And then see where it takes me.
Well, to wrap things up then, I guess the last question, which relates to what we talked about, maybe a different answer, is if you could abolish one misconception in science and get rid of it permanently, what would it be?
I don't know if I would use the word abolish, but I think if there was one thing which most of us could get over, right? I think from the time we were little as kids and we were reading books about scientists, we see Einstein, we see his mad scientist hair, and we put all of them on a pedestal. We're like, oh, all these scientists are geniuses, which I'm not saying they aren't. they're geniuses. They're all very smart people. But I wish people wouldn't put scientists up on that pedestal, because then it makes it seem very hard to reach for a lot of us. A lot of minorities, especially, again, people who've come from backgrounds where their families, they're first generation college educated, they're first generation, professionals. And they just look at scientists and they put them up on a pedestal and say, oh, I'm not even going to try because they're geniuses. And I think I would like to remove the misconception that intelligence is one thing and that a high IQ defines being genius. I think there's so many concepts or so many varied aspects of intelligence. For example, emotional intelligence. And there's so many things that make you a successful scientist, that you don't realize what you're good at, that someone else might not be. And I don't know if, what we have seen scientists of the past who had locked themselves up in rooms and were geniuses scribbling equations on boards would be successful today, where you need to go out there, advocate for your science, be good at scientific communication, get your ideas across to grant reviewers, across to other scientists, get the grant funding, be able to collaborate, again, go out there, establish successful collaborations to do the science. Again, be someone who's fun to work with so that other people want to collaborate with you. All of those are aspects of success. All of that makes good science. Being able to pull aspects from other fields, from other kinds of studies, apply it to your own, to answer questions that you have not been able to answer before. It takes the field forward. So I wish people wouldn't put scientists up on those genius pedestals and say, oh my God, they're just up there and I can't reach them. Because understand there's so many different aspects of intelligence. And if you want to do it, you can do it.
I like that. I mean, absolutely agree with that, because in the end, if you want people to become more involved with science, also you have to make it more accessible.
Exactly.
Yeah, for sure.
And saying scientists are geniuses, in a way, again, they might be smart. I'm not saying they're not. It makes it inaccessible in a lot of our heads.
Yeah. So on that note, we thank you so much for joining us today, Dr. Priya Devarajan from Stony Brook University, today at the Immunology Podcast. It was a really nice conversation. Thanks for joining us.
Thank you, Brenda. Thank you, Jason. It was really fun. I have a lot of fun talking about science, a lot of fun talking to the next generation of scientists. And like Brenda said, if any of you are interested in working with me, go check out my lab website, www.thedevarajanlab.com, or you can look up my profile at Stony Brook University and we'll lead you to my website. And I would love to hear everyone's feedback, ideas that people have, be it an undergrad, a high school student, anyone can do good science and everyone has great ideas.
Excellent. Well, it's been a pleasure.
It was really fun. Thank you.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup Papers. You can also reach out to us on X at Immunopodcast or via email at info@immunologypodcast.com with feedback or to suggest a guest. See you next time.